To kick things off, please welcome the CTO of Ginkgo Bioworks, Barry Canton.
Good afternoon, everybody. I am very lucky in this afternoon session. I'm going to, as Jake says, I'm going to get to tell you a little bit about the technology we use at Ginkgo and also the scale and the multimodality of the data that we're able to generate. To share that with you all, I'm going to be joined by 4 incredible technical leaders from across Ginkgo. One of the themes that we're going to touch on today is how we combine scale with flexibility. For our partners, scale means that we can work at high throughput, and the flexibility means that we can customize our workflows to suit the specific needs of the project. For Ginkgo, combining scale with flexibility means that we can run many programs across many market verticals and drive our scaling flywheel. But before we get into all of that, I want to spend a minute talking about why data is such a big bottleneck in biology. My guess is I don't need to convince that many of you about this, but I am going to give 3 examples that I think are interesting and impactful. The first example is that there are just many fundamental things that we don't know about biology still today. Researchers, this is on the left here, researchers at JCVI took a very simple microorganism and they made it even simpler, as simple as possible. They eliminated all of the genes that were inessential for the cell to live. What they were left with was a simple microorganism with approximately 500 genes that are all essential for life and that are highly conserved. They occur everywhere. You would think that we would know what all of those genes do. In reality, there's about 20% of those genes that have unclear function. And as you move up to the layers of the chain of complexity of life, the situation doesn't get better. There's still huge fundamental things we don't know. So that's kind of the underlying cell. My second example, let's look at what happens when you go to engineer a specific system. And you'll hear more about this system a little bit later. Chimeric antigen receptors, or CARs, are sort of the foundation of cell therapy. There's a huge combinatorial design space here, as there is in almost every domain of biology. So a CAR is composed of a number of domains. We'd like to be able to optimize each of those domains individually, and we'd also like to be able to find the optimal combination of domains. To do so, you need to look at hundreds of thousands of different combinatorial designs. And the real kicker is that you'd like to be able to do that in a context that's relevant, a disease model that's relevant to understand a challenging tumor environment. Third example. Everybody is taking as much data as they can find, and they're using it to train AI models, which we hope will solve big problems. We should absolutely do that, and we all are doing it. But a finding that is common across technology and biotechnology is that while we have really impressive AI model technology, and we have incredible compute, thanks to partners of ours like Google, models tend to be undertrained because there is a shortage of the right data in order to be able to train them effectively. AI has really thrown a focus on this data shortage, but at Ginkgo, we were worried about data before it was cool to be worried about data. This is something that we have been thinking about for a long time, as long as the company has been around. What we have been building at Ginkgo, our Foundry that many of you have seen or have heard about, is really, you can think of that as being a data engine. It's a collection of capabilities for generating high-quality, scalable data that is deep and that is multimodal in nature. And throughout Ginkgo's development, we've made choices to invest in measurement and testing technologies that give us data that is rich and really allows us to see what's happening inside of the cells. We take this collection of data capabilities, some of which are shown up here, and we use them in a variety of ways for our partners. For example, with lab-in-the-loop workflows, where you're going from model to data and back to model again, or where we're collecting large multimodal data sets for pre-training of models. And now we're going to be using these capabilities for lab data as a service, as Jason announced this morning. We're able to do that because of an underlying set of capabilities and technologies, including automation, including our biological code base, AI and ML modeling abilities, and also operational excellence. And all of those taken together, they give us a scale and flexibility that allow us to help our partners with data, with product discovery, manufacturing, and making manufacturing cell lines. Today, we're going to highlight one of these technologies, and it's an automation technology that kind of rethinks how lab automation could work. And I'm very lucky to have the best person to be able to tell you about that, which is Ginkgo's head of automation. This is a technology that was developed at Zymergen and is now being further developed and deployed at Ginkgo. So please, Will Serber, if you want to join me and take it away.
All right. Thank you, Barry. So I'm here to tell you a little bit about our proprietary automation technology, an example of which you can see over on the side of the room. And first, I want to motivate why we decided to innovate in this area in the first place, because that might not be obvious. So I would state that we need high throughput, high quality, and very flexible automation in order to engineer the diverse organisms that we work with and to create the volume and richness of data required to build foundational models. So the challenge really is that you cannot buy this off the shelf. To tell you a tiny bit more about that, it is very common to have a paradigm where you have basically one work cell corresponding to one process or one workflow. And one of the fundamental reasons for that is there's generally in the automation continuum a trade-off between flexibility and scale. So if you want maximum flexibility, you'd be over on the manual side of things. And if you want maximum scale, you'd be on the integrated work cell side of things with sort of walk-up instrumentation in the middle there. Now, we found this to be an unpleasant sort of trade-off to make, and so we set out to make something that was flexibility at scale, which is a fourth level of automation, which you see here, and I think we succeeded at this. The solution that we came up with is a technology, a modular automation technology that we call racks, or reconfigurable automation carts. The traditional work cell is really a big central arm with a bunch of instruments arrayed around it. And you can see that this architecture is very different. So in this case, we have an enclosure with an instrument inside, and it's got a small 6-axis robotic arm that is feeding just that instrument. And you have all the things, the sensors, the electronics, the data in, the data out, all that sort of enclosed within that enclosure. And so it becomes this little self-contained Lego building block, if you will. If you put together a bunch of racks, you get a rack system. And you can imagine we spent a lot of time thinking about how to make a system that was easy to reconfigure and easy to build. So the system we have over here arrived in crates from California the other day, and we spent maybe about 3.5 hours getting it to the point where it was actually fully running, which I can assure you is very fast relative to state-of-the-art in the industry. One of the really nice things about having this Lego-like approach is that you can scale very well. So you can make very small systems with, say, 5 racks. You can make really big ones. So here we have ones that we designed that is 185 connected racks for high-throughput phenotyping. There is no other technology that could make a system anything like this. So I'm focusing mostly on the hardware, but we spent, honestly, probably more effort on the software than the hardware. It's just a little less fun to talk about, but I'll tell you a bit about it. So here we have a display of the work cell, the rack system level view. And what we have is, at that Gantt on the top, every instrument has its own row. The red line moving along is time. And then every colored brick is an action occurring on an instrument. One of the things that might not be obvious, so if you had a really big rack system, it would be very painful if you could only run one workflow at a time. So we put a lot of effort into our scheduler and enabling running of multiple workflows at the same time on the same system. So the different colors there, the blue, the purple, the green, are totally different workflows running on the same system. It is not just cool technology, it is also meaningful in terms of the value delivered to the organization. So as we've gone from level 3 automation, which is to say traditional work cells, up to racks, we have commonly found that we can reduce the labor time by as much as 80 to 90%. Sometimes we are able to have a big impact on cycle times as well. So for example, if we can automate across a team handoff or overnight, we've gotten cycle time reductions of as much as 60% going from level 3 to level 4 automation. One of the less clear things, the last point on this slide, I'll read out loud. So heavy parameterization entirely changes protocol development and use is especially non-obvious, so I want to dive into that a little bit. This is an example of the onboarding of a qPCR workflow onto racks. This is the same process that we would take if we were onboarding onto traditional automation. And you don't have to worry too much about the details. The main takeaway is that it involves a lot of teams, scientific teams, operational teams, and high-tech teams, to not just write the protocol, but also to sort of set up the entire stack upstream and downstream of that so that the operators are getting the right instructions, we're getting the data off, and it all just works seamlessly. And this is a very painful process. Now, we onboarded qPCR, the first couple qPCR protocols, and this worked just fine. But our own automation control software allowed us to take the next step, which you couldn't do on traditional automation, which was we made a generalized, highly parameterized version of the qPCR protocol that let us cut out the vast majority of these teams. So instead of those sort of 5 teams, now it's a program team that is just passing parameters to an operational team, and they can run those workflows, and all the data and all the instructions just sort of comes along for the ride. So this is a transformative change. This leads me to my conclusion, which is that flexible automation drives better scientific outcomes and produces richer data to fuel AI and closed-loop systems. The traditional lab automation view is that automation is useful for capturing lots of data once you have lockdown processes. And we would like to change that and instead suggest that the best automation accommodates variation and experimentation to produce far richer and more relevant data sets. To leave you with an analogy, traditional sort of manual lab work is a lot like monks transcribing books by hand. It's very error-prone and very slow. The traditional lab automation, the traditional work cells, is a lot like a printing press where you are doing all the typesetting. So it works to produce many copies. It's very high throughput, but it is very painful to change. Whereas what we've made is more like a digital laser printer. So you can produce not just high throughput, but also many different works. And if you imagine ChatGPT, it doesn't need 1 million copies of Moby Dick. It needs 1 million different books to learn from. So as we build foundational models in biology, what we need is that ability to have highly parameterized, highly variable workflows. And that's what we've provided. Thank you.
OK, thank you, Will. So next, we're going to hear 3 short vignettes from 3 technical leaders at Ginkgo from different parts of the company, working in different domains and using different kinds of data and generating it in different ways, with robots, with pooled approaches, modeling with AI, and even doing in vivo discovery. First up is going to be Emily Wrenbeck, PhD, who leads protein engineering at Ginkgo, and she's going to tell us how we integrate experimental data with AI modeling to make better enzymes for our partners. Emily?
Thanks, Barry. Yes, so one of the long-standing challenges in protein design is that protein sequence space is enormous, right? It's something on the order of 10 to the 35 possibilities. Right, so the key question is, how do you navigate such an enormous search space, right? And in a more real-life, practical scenario, when you have a specific protein sequence in hand, along with a list of, let's say, hundreds of known beneficial mutations to that sequence, how do you smartly recombine those mutations in a way that enables you to take bigger jumps in protein diversification and thus performance improvements? Another challenge is that the sequence to function relationship is something that has to be learned. And especially for enzymes, the learnings from one problem space to the next don't necessarily translate. And further, context is extremely important, right? Things like temperature, pH, expression host, et cetera, for determining what the shape of those sequence to function relationships looks like. And so at Ginkgo, we've built a platform that enables us to use experimental data generated in the Foundry to iteratively improve enzymes with a machine learning guided design process. And so we typically start by first screening a library of natural orthologs, which we mine from both public and proprietary sequence databases. And in this process, we always typically discover much better starting enzymes as points for engineering further. We'll then take these lead enzymes, and we'll design a first-round protein variant library using all the latest and greatest protein design tools, including some of our own. This library is then screened in the Foundry, and we'll get back a data set that has that sort of sequence to function information. We can then feed this data set into supervised model training, and we can leverage what those models have learned to then help us design the next round of sequences. And this process can be iteratively repeated until we hit some target metric. And so in practice, what we observe is that this approach is incredibly effective across both diverse and engineering objectives as well as enzyme classes. And so in these plots, what's being shown on the x-axis here is the round of engineering. On the y-axis is the activity fold improvement shown in teal. And on the secondary y-axis is the library size at each round shown in gray. And so the general trend is an uptick in performance round over round, right? And in this particular highlight case study, we observed a remarkable 80-fold improvement in 3 rounds of iterative machine learning guided design. Another trend that we often see is that in later rounds of engineering, when sufficient data has accumulated and the model performance prediction gets really good, we can often take big jumps in performance in very small library sizes. So in that particular case study, we were able to make a leap from 6 to 10-fold improvement in a library size of only 10 members. And the final point here is that what the machine learning really helps us do is make that smart recombination, right? Both within the space, but also to new spaces. And so these purple bar charts here show an average mutational depth at each round of engineering. And again, it's just really to help us take those bigger leaps in protein diversification and thus performance improvement. So I'm super excited about the consistency of the effectiveness of this approach across both those problem spaces and enzyme classes. And I just want to finish and close by underscoring that it's really all fueled and enabled by the data that's generated in the Foundry, both by those super cool robots, go check them out, but also our exceptional experimentalists. Thanks.
Thanks, Emily. Next up is Uri Laserson. I'm very excited about this. So Uri just joined Ginkgo. Until very recently, he was the CTO and co-founder of Patch. He's going to tell us a little bit about the work that they did at Patch, and you'll see how naturally it fits into the Ginkgo platform, I think. So Uri, please take it away. Thank you.
Thanks, Barry. So I'm going to tell you about an approach that we have been taking for discovering new RNA elements that can increase the stability of an mRNA molecule, and therefore also increase the protein output of the mRNA molecule. Now, as Emily said before, the space of all possible sequences is astronomically large, and most of it is non-functional. And so what people typically do when they're looking for functional sequences is they'll limit themselves to the natural genome, and they'll screen natural sequences to find something that basically works. The problem is that evolution is not optimizing the same set of traits that a biotech company cares about when they're trying to design an RNA therapeutic. Really what you want to do is to understand what is the underlying regulatory grammar for how the mRNA is being stabilized. The way that we're doing that is by combining very high throughput pooled assays with the latest advances in AI. So the way that this works is you start with, say, 10,000 candidate sequences that you think might be interesting, and in a single pool, in a single tube, you go and you synthesize them, and you clone them, and you transcribe them into mRNA, and you deliver them into cells, and then you take time points of those cells over several days. And then you use next-gen sequencing to quantify the abundance of all of the individual mRNA molecules all at the same time. What you end up with is a data set like you see on the right, where every single one of these curves is a unique mRNA that we designed. And over time, on the x-axis, what you can see is that they're all degrading, as you would expect. But crucially, they're all degrading at different rates. And so what you traditionally might do is you would pick a dozen of these sequences, the best ones, and then carry them forward in your development pipeline. In contrast, what we actually do is take the entire data set and train an AI model that learns how to map from any given input sequence what is the predicted stability of this sequence. Now, this is the model that's actually going to kind of understand something about the underlying regulatory grammar. And most excitingly is that now what we can do is take tens or hundreds of millions of design sequences and crank them through the model and figure out what is their predicted activity going to be. From that huge set of sequences, you can now take another 10,000 set of sequences and then go synthesize them and assay them and generate a new data set, and then use that data set to update the same AI model. And then you go and you do it again. And the idea is that for you turn the crank a few times, and for any given set of therapeutic design constraints, you can find synthetic sequences that are going to be better than natural sequences. The coolest thing is that this actually works when you translate this into a mouse model. And so what you're looking at here is we've taken like a commonly known benchmark UTR sequence and one of our AI design sequences, and we've plopped them into an mRNA that is driving luciferase, and then we package it up in an LNP, and we inject it systemically into a mouse, and then we take time points over 2 weeks of that mouse to see how much protein is actually being produced. And what you can see in the curve in the middle on the bottom is that the difference between these two curves is strikingly different. And actually, we can see that the AI design sequence is driving expression at higher levels and much longer. And we even see fold changes that are up to 100x stronger than the commonly used benchmark. So that's it.
Thank you, Uri.
Thank you.
OK, we're excited to have the patch technology deployed across the Ginkgo portfolio of programs. OK, last but not least, I'm excited to introduce Shawdee Eshghi, who was one of the first mammalian cell engineers to join Ginkgo a number of years ago and build out our platform, and now leads a lot of programs in cell therapy. And she's going to pick up the chimeric antigen receptor story that I introduced at the start. Shawdee, please take it away.
Thanks, Barry. It's great to be back up here to share with you a little bit about how we think about generating large data sets across our biopharma programs. And in particular, the relevance of the model systems in which we're using to generate the data. If you think about the ChatGPT example, again, it wasn't trained on random collections of words and sentences. It was trained on relevant words and sentences from different styles of writing, like Jane Austen or the New York Times. And when we're asking biology to report on what's happening in a complex human disease, we need to get data that's as relevant as possible. And so one example of how we're thinking about this is from our CAR-T story that Barry started out with. We've gotten really good at screening these large libraries in vitro and lots of different tumor cell lines. But we wanted to even push that a little bit more and ask, what is a more relevant model we can use? Can we do this in an actual tumor-bearing mouse? And together with our partners at the University of Wisconsin, we did just that experiment. This is a clinical group that is studying neuroblastoma, which is a devastating pediatric cancer, and they're targeting GD2. And so we built a library of 10,000 chimeric antigen receptors that also targets this antigen. And to be honest, I was really skeptical about this experiment. It's really hard to work with large libraries, even in simple controlled settings like cell lines. I really didn't think we were going to get this kind of positive preliminary data shown here. So on the slide on the left, right, is tracking the barcodes, which report out on each individual CAR design. And so our theoretical maximum is 10,000, and we maintain most of that diversity all the way up until the time that we inject the cells into the mouse. And 23 days later, after interacting in the tumor environment, we're recovering 1,000 barcodes, suggesting that there's 1,000 different CARs that have persisted in this complex environment. Now, I've done enough animal studies to never trust one mouse, and so we wanted to look at how many designs are shared across replicate mice, and that's what's shown in this graph here. And remarkably, there's a number of designs that are conserved across 3, 4, even 5 mice, suggesting that whatever selection is happening there in the mouse is real and not random. And so we're now following up on these candidates and evaluating them as potential therapeutic candidates for our partners at Wisconsin. And so we're doing this kind of pooled in vivo work across our biopharma programs in gene therapy and RNA and gene editing as well. But there's some scenarios for which even an animal model isn't good enough to generate large data sets. So to take one example from genomic medicines, like CRISPR therapies, you need the human genome there to really assess the safety and efficacy of any kind of genomic medicine, and a mouse is never going to do that. In another example, mouse tissues are very small. There's a small number of cells, so you can't accurately screen a large library. And these are scenarios where human stem cell models that we can use in vitro can recapitulate some complex disease states. So what's shown here is a diagram of a human choroid plexus organoid. This is a brain tissue where the cerebrospinal fluid is generated, and it's an important target for gene therapy. And so we've generated these organoids in our labs at Ginkgo, and we're going to be scaling this and using it for screening promoter libraries for gene therapy applications. So as much as everyone loves data, I would challenge you to think about the relevance of the data. And while we do use simple model systems to build our methods and scale them, we are always thinking about how to push them to more complex models. And that's it.
Thank you, Shawdee. I hope you'd agree that those were some amazing examples of what we can do with the Ginkgo platform. It's here. We're able to do amazing things in enzymes, in RNA, in cell therapy, and beyond. We look forward to helping you with your next program to generate data and to solve some real problems. So thank you for today, and please enjoy the rest of Ferment, and we'll see you at the break.